Hello, everyone. I hope you all had a great time attending our two sessions today. The first session was Astrochemistry and Biosignatures, and later there was a session, Synthetic Biology and Extremophiles. If you missed any of the talks, you will be able to watch them in the replay zone on Hopin after the event has ended, so after the last session. And now I am happy to welcome our key speaker, Professor Sara Walker. Sara is an astrobiologist and theoretical physicist. Her work focuses on universal laws of life. These laws could guide us in search for life that we don't know it. She works at Arizona State, where she is Deputy Director of Center of Fundamental Concepts in Science, and also Associate Director of Center of Biosocial Complex Systems. She's also Associate Professor in School of Earth and Space Exploration. And last but not least, she is also very active in public engagement in science. Sara, it's great pleasure to welcome you here. And when you're ready, please go ahead with your talk.
Excellent. Thank you so much. I'm thrilled to be here and to be a part of the first meeting. So can you see my slides full screen now?
Yes.
Excellent. Okay, great. Yeah, so thanks so much for inviting me. And I'm excited about talking about some of the ideas on how we might get at deeper principles about the nature of life and how that can help inform what we're doing in astrobiology. And since, this is sort of a new endeavor, having this Astrobiology Society, I thought maybe I would start with talking about sort of a vision for astrobiology in the next 100 years. And this vision is clearly biased because I'm, as Tomas said in the introduction, I'm trained as a theoretical physicist. So I tend to be interested in deep abstract principles. And it may or may not be the case that that's the right way of thinking about the problems we have in astrobiology. But I find that looking at the history of physics, at least, there's a lot of promise for the opportunity that we might actually uncover universal laws that apply to living systems in the same sense that we were able to, say, find universal laws that apply to gravitational systems or any other systems that we think that we understand universal principles of. So, for example, like quantum systems or the Standard Model. So just, I think it's incredibly instructive to look sort of at historical sequences of events and how the history of science has played out. So just thinking about, the history of maybe one of the greatest ideas that evolved in physics in the last 100 years, Albert Einstein, in the early 1900s was coming up with his ideas about the nature of space and time by starting thinking about the properties of light. And it's really interesting to think that he formulated the ideas of space-time and the fact that it should be curved to explain gravity over 100 years ago, but also sort of in the same time period, at least in the United States, like just to give you kind of a mental picture of what was happening. I think it was just like the same year that there were more cars than horses on the streets of, at least in America. So this kind of gives you just an idea of how long ago that was and how our technology was at the time that he was thinking about these ideas. And in fact, his ideas were radically more advanced in terms of their depth and their explanatory power than explanations that we had, say, a few thousand years before. So the way people used to explain planetary motions in ancient Greece was in terms of epicycles, which were circles inside circles, and the Earth was the center of the universe. And basically, they had a really good predictive model of why planetary motions were the way they were, but it wasn't explanatory, and it wasn't something you could extrapolate to other star systems or use for anything other than the exact intentional purpose it had, which was to explain planetary motion. And of course, Newton came up with the idea of gravitation in the first place that Einstein built on. And eventually the understanding we came to was this kind of deep understanding of the nature of the actual reality we live in, that, none of us would sit here and intuitively think that we're sitting in our chairs right now because the space and time around us is curved in the presence of massive bodies. So this is, obviously a really profound insight into the nature of reality, but it also made incredible predictions of phenomena we could never anticipate without that kind of deep knowledge. And so what I've shown on the right here is just a picture of one of the Ligo detectors. Ligo is the large telescope. Basically, it's an interferometer that was designed to detect gravitational waves. And this was a multi-decades long project. Each arm on the interferometer is 4 kilometers long. And that's necessary so that you can measure with a precision that's about 1/10,000th of a proton's width. Right, so this is a huge instrument to measure really tiny and very precise measurement. Some speculate that it's actually the most precise measurement we've ever made. And it was successful in detecting the presence of gravitational waves, confirming their existence almost exactly 100 years after Einstein predicted that they would be there. So this is a phenomena that we would never be able to interact with directly with our senses. It's something that exists in our universe, but we had to use theories to predict, and we had to use those theories to guide instrumentation and all of the kinds of things that we actually had to go through to be able to detect that phenomena in our universe. So that's sort of a 100-year history from Einstein's idea to actually building the devices and the technology to infer or make first contact, if you will, with gravitational waves, but we want to make first contact with aliens. And in astrobiology, I think we're in a really different situation because we don't have any guiding theory yet. So what do we have when we're talking about sort of ideas about the nature of life? Where were we 100 years ago? Where are we now? Where should we feasibly be heading? Astrobiology 100 years ago. So there was, I'm talking about original life astrobiology here. There were other ideas about the search for alien life. Like people thought 100 years ago that there was life on Mars, for example. But this quote is actually from a book. Well, it's an essay by Haldane, who was working at the same time as O'Para, and they came up with this idea of like the primordial soup hypothesis, which is that on early Earth there was like a chemical mixture, and it just became complex enough to become life. And that actually was a very forward-thinking hypothesis about the nature of life. And we've been working on related ideas for the last 100 years. But a lot of the sort of premise of most of the original life work we're doing is that life is chemistry. So if we just build a complex enough chemical system, we'll solve the problem of life. And so there's some convolution between the concept of what we think of as life and what we think of as the molecular components of life. And it's a good starting place, but one of the questions I have is how can we actually push that further, and how can we think about deeper abstract principles? So when I look at sort of, the complex chemistry of life and the mapping of the molecules and the reactions, like what's shown here on this slide that maybe metabolism uses, it seems to me a bit like an epicycle model. It's very predictive. It's very descriptive of that exact system, but we haven't built ways of generalizing beyond what we know. And of course, the big problem is we don't know what life is. So these are just some common keywords that are used in definitions for life. And you can just see that there's a lot of recurring motifs, replication, information, metabolism. So all the sort of buzzwords you might see in descriptions of what life is. People have come up with various definitions for life. This is one that was proposed by Carl Sagan in a really nice short essay. It's only 2 pages. I highly recommend it. It's just very clever. Where he says life could be defined as any system capable of performing a number of functions like eating, metabolizing, excreting, breathing, moving, growing, reproducing, and being responsive to external stimuli. So this seems fairly reasonable. But Carl was a little cheeky in this essay, and he pointed out that any aliens using this definition of life that might visit us would probably think that cars are the dominant life form. And probably even nowadays, they might really think that because even cars have been to space. So we have this kind of paradoxical situation where definitions for life that we might give these kind of counterexamples that maybe people don't feel comfortable with. So Carl's point with this example of this definition that seems reasonable, and then pointing out cars would be considered alive by this definition, was to say that nobody wants machines to be alive. And I would actually disagree with that, because I think our technology is a manifestation, as much as we are, of the physics underlying life. And that really, seeing that picture clearly really requires going to this deeper level of abstraction. So we don't have definitions. And a lot of people have made the argument that what we need is not definitions, we need theories. So for example, Carol Cleland, Chris Chiba wrote a wonderful paper about defining life many years ago, making this argument. And so a lot of my time is really spent trying to think of what would those theoretical principles be and how do we actually think about it. And I just want to point out that the situation is sort of so bad, especially when you're thinking about the chemistry of life and how you organize origin life experiments, that people really talk about the fact that life is not actually even a property of molecules. So this "life does not exist" quote actually came from Andy Ellington. I'm sorry, I don't have the reference. Got highlighted out of this slide. And I recall him saying that at an American Chemical Society meeting. It was actually on one of his slides, just to say, thinking about life is not a useful concept when you're trying to build the molecules of life and talk about engineering the origin of life. It's just kind of an irrelevant concept at the scale of individual molecules. This quote from Jack Szostak is a little bit more eloquent because he talks about how when you're thinking about a defining property of life and you start looking at the small scale of molecules, it seems to be that, those properties kind of disappear or they just blur out. And so this, of course, if you want to think about it the other way, you can just say, well, life's an emergent property. It's not a property of individual molecules. But again, we get to the situation where, well, what is that emergent property and how do we actually talk about it? So these are sort of some of the debates and things that people are having and ways of thinking about it. I'm really, as I mentioned, I already kind of, I have to state my biases as a theoretical physicist because I am very interested in deeper principles. And it could be the case that, say, Andy Ellington's quote, "life does not exist," is actually true. And life is just not a natural category that exists in the universe, in which case we have to think very carefully about what we're doing as astrobiologists because we're not looking for a universal phenomenon in that case. I do think that life is a universal phenomena. I think it does have universal features, but I think that those are probably very abstract in the same way that the sort of curvature of space-time is abstract and not directly amenable to our perceptions. Life, one of the reasons it's been difficult to define is we're looking at life on Earth as specific instances of something that's a much deeper phenomena that happens in our universe. And we want to be able to explain those features from a deeper story. Where that sort of tradition comes from, a lot of people have been asking this question, what is life? So I have shown here a picture of Erwin Schrödinger, another famous physicist of the last century, famous for his work in quantum mechanics, but he also wrote this wonderful little book called What is Life that a lot of people cite and talk about, where he asks about how we can actually account for what happens in a living organism by physical or chemical principles. And he admits sort of toward the end of the book, and I just absolutely love this quote because I think it's very, forward thinking about the nature of the problem we have to solve, where he says, living matter, while not eluding the laws of physics as established up to date, is likely to involve other laws of physics hitherto unknown. And so I say this is sort of life is what? Like the answer is actually really going to be very surprising to us in the same sense that you couldn't have told, say, the ancient Greeks that the planetary motions were actually because of this space-time curvature. So I think these analogies about thinking how abstraction and theory building has helped us in the past in science are actually really useful for astrobiology. And this point about it's not eluding current laws, but might involve other laws, I think is also very important. So just back to sort of Einstein's perspective on things. This is from an essay that was in a book honoring Max Planck on the occasion of his 60th birthday, that we might have general laws on which the structure of theoretical physics is based, which should apply to any natural phenomena. And those laws should actually also apply to life. So obviously, like gravity applies to me as a living thing. I don't defy the laws of gravity. But can we actually get something that explains me in the same way that I can explain why I'm sitting in this chair right now, but explains why I'm here at all and the evolutionary process the universe underwent to create me or you. So where I sit on this is kind of a very abstract and big picture way of thinking about things. And it's just kind of a current snapshot because these ideas are a moving target and they change all the time when you're trying to come up with new ideas about the nature of life and how we think about it. But if we think about sort of classifications of current physics, what they apply to, what they describe, and then what might be this sort of missing physics that we don't yet have to describe life. We're pretty good at describing the properties of matter and light, and that came from quantum mechanics, and the Standard Model is an excellent example of that. Space and simultaneity came from special relativity and general relativity. So obviously we have this concept of space-time I was mentioning, and large numbers and observational uncertainty. I mean, we have this theory we call thermodynamics and statistical mechanics, that applies to many body systems with certain kinds of properties associated with them. And we can talk about how those systems behave on average and do all of these kind of things. What my gut feeling is, which may or may not be right, but this is sort of, so I've set up so far in this talk that we should be thinking at sort of this deeper level as astrobiologists to be really able to get to the answers to the questions that we're asking. That may or may not be the case. But I think it's a research program that we really need to take seriously, and we need to start investing more time and effort into. And then when you look in that, you know, what kind of theories would you construct, and how do they couple to experiments, and how do we think about deeper principles? There's a particular subset of ideas that I'm really interested in and have been working on because I think they're the most promising. But again, you know, it could be there's some other idea that somebody's brainstorming somewhere now that could really be revolutionary in this field, and it has nothing to do with what I'm proposing. So this is wide open. It's exciting. And we're just really trying to figure out the nature of ourselves. So for me, I think the thing that is most intriguing about life that doesn't really fit cleanly into the other laws of physics is really this idea of thinking about what information is in physical systems, and how it is that certain things come to exist on certain planets. And I'll explain a little bit more about what I mean by that in a minute. There's a few theories that I think are trying to get at this. So I work personally on something called assembly theory, which if I have time at the end, I'll get into a little bit of detail on. And then there are some related ideas in what are called causal set theories for quantum gravity, which have a kind of a similar structure, which is kind of an odd connection to make. I don't know if I'll have a lot of detail to go into that. And then I also am a huge fan of what's being done by David Deutsch and Chiara Marletto in constructor theory, which has some also related concepts. So what do I mean by focusing on information and what is possible? I'm going to give a couple really anthropocentric examples, but the sort of logic applied to these examples can apply equally well to us as a technological civilization as they can to what cellular life invented in terms of information processing systems and the capability to transform matter in novel ways that are statistically impossible to occur in the absence of an information processing system. And so that's sort of the key point. So what I'm showing here is just some features of technology that are processes that happen. They're consistent with the laws of physics. In fact, you have to have a knowledge of how the universe works even to mediate these processes. So you have to be an intelligent being like we are. And just thinking about, you know, sort of how that opens up the possibility space of what the universe actually can do. So one example is, you know, our planet is the only planet we know of that's anti-accreting on a regular basis little metal boxes into space. We launch satellites, you know, almost on a daily basis. In order for a planetary surface to be building boxes and throwing them into space, you had to have some kind of physical system on the surface of that planet, i.e. us in this example, that over its evolutionary history acquired knowledge of the laws of gravitation and the engineering principles necessary to actually implement this kind of transformation. So planets usually form by accretion. They accrete mass, but they don't usually throw mass off into space or even send it to other planets. We're doing that. And then the other one here is just the periodic table of the elements. It's showing sort of all the physical formation mechanisms for different elements in the periodic table. And when you get to these very, very high atomic number elements, right now we're all the way up to 118 on our planet. The most reliable process for making those in the universe is technology. So nuclear physics allows those things to happen, but what makes them happen, what actually makes them physical and makes it so that you can make one of those elements in high abundance is technology. So you have to have physical systems that have knowledge of nuclear physics in order for those elements really to be produced in a reliable manner. And so the sort of key feature here that we're interested in is what does it mean when you have a physical system that has information or knowledge, making more things possible or even more things probable? So improbable structures that don't really exist in the universe outside of life are also, you know, the molecules, the macromolecules that we see inside cellular life. They also require information to specify their formation. They don't occur spontaneously in the universe. If we did, we'd have original life events all the time. But they require a process of evolution, acquisition of information, and the ability to construct those objects. And so the argument that I'm really interested in are the sort of set of ideas to try to get at these deeper principles is to understand life generically. And I mean life from everything from the origin of life transition all the way up to what we're doing now, even by me giving this talk right now, it's a manifestation of living physics. All of that is described by the part of the physical world where information becomes a really dominant participant in the kind of physics we're talking about. And so just to go back to sort of the analogies from physics, because I think they're helpful, you know, I've been using gravity a lot. Gravity is universal. It applies everywhere in the universe, but we don't talk about gravity as being a dominant or relevant physics inside an atomic nucleus because it's too weak. Where it does matter is on, say, a planetary scale or a galactic scale or on the size of the whole universe. Information, likewise, I think is a universal physics, but it doesn't matter to describing the system that you're interested in until you get into this high combinatorial space where you have an explosion of the number of things that could exist that you actually need information to specify what exists. And this happens in chemical space. So if you think about all possible molecules, it's actually exponentially huge, and it's so large that you don't even have enough resources in the entire universe, say, to make all possible proteins of length 100 amino acids, right? So there's a very small, like if you think about small molecule chemistry, there's this combinatorial explosion. So if you want to say specific things out of that universe of possibilities exist, what I'm suggesting is that there's a new physics that emerges there, which is what we call physics of life, or what we call information. And that's why we exist in that very high combinatorial diversity space, that high complexity space. So the theory that I've been working on with colleagues, so this is mostly in collaboration with Lee Cronin's lab at University of Glasgow, and we have probably about 10 people between our groups working on developing ideas related to this theory, is called assembly theory, which is really just getting at this idea that if you take an object that exists in the universe, say a molecule, you can talk about all the possible ways of forming that molecule by breaking it apart into parts, and then you can look at the pathways for producing it. And the argument that we make is if that path is sufficiently long, the combinatorial space where that object exists is too large, then you would require a living process to get there. You would require a system that had information about the specific steps. And so part of the idea here is to really think about life as a continuum. And there's some physics that exists in the space of all things that are possible that talks about the causation between different objects and how objects get built up from other objects, and why when we see a high complexity object or a high assembly object in assembly theory, it's actually a biosignature. And I'll talk about that a little bit toward the end of my talk in more specific ways in terms of the context of how that theory builds into things we can observe. So I want to go through some technical results from my group and some of the stuff we've done in assembly theory in a minute. But I just wanted to go back to this sort of big picture. I was thinking about you know, this idea of gravity over 100 years. What are we going to do in Origins of Life, and how do we do that? So one of the things is the theory has to be coupled to experiments, and also has to be coupled to observations we might do with space telescopes. And so there is a lot of work being done in sort of systems chemistry approaches, and also building robotic systems that can explore, say, much more of the parameter space that might exist on planets, and allow this sort of recursive exploration of chemical space to look for this kind of physics that we're describing. And so you can imagine this is kind of being, you know, the very early interferometers that were built in the 60s, you know, were very tiny. They couldn't really actually explore enough to see gravitational waves, but they were sufficient to sort of demonstrate the kind of principles we might need to get at doing that in subsequent decades. And really sort of my holy grail of what I'd like to see for astrobiology and origins of life, which is deeply connected to how our ability to look for life on other planets, is to do the kind of massive international collaborative experiments that we do in physics. So this is just an example of the Super Kamiokande experiment in Japan, which is looking for proton decay. And they have huge volumes of water looking for a very rare event. You can imagine if we had a really good guiding theory telling us what life is and how we should look for it emerging in undirected chemical systems, that we can evolve through these sort of robotics, you know, exploration of chemical space. And we built, you know, a large experiment to explore as much of chemical space as possible. Would we actually find those regions in that combinatorial space where new life forms actually emerge? So can we look for alien life on Earth by building experiments basically at a scale that actually allow us to explore all the chemical diversity that our universe actually can generate under different planetary conditions. So that's sort of like the big vision. I took a little longer with that than I wanted to, but I think setting the stage of like where we're going with questions is really important at this stage in astrobiology because we're such a new field. So I'm using that more as a sort of inspiration, like let's all start thinking a little bit more about connecting all these pieces and how do we actually connect it to explanatory theory? Because for me, that's really the thing that astrobiology has been missing is a real emphasis on what is the phenomena we're looking for and how do we talk about it in a rigorous quantitative way. What I want to transition to now for the second part of my talk is really sort of some of the work that my group is doing now and how it kind of feeds into that sort of bigger picture vision. So what can we do now? If you were thinking about this 100-year vision for astrobiology, ideally we could compress that into 20 years or something. So most of us here can, actually see these things happen in our lifetime, but I think it's hard to do. So my group works in a lot of areas, trying to kind of converge this sort of big picture vision into like concrete things that we can work on. I'm not going to go through all of these now. I talked a lot about sort of thinking about life as a physics of information causation at a philosophical level. We also do a lot of work in that. And I am going to talk about statistical approaches to characterizing life's chemistry, which is also deeply related because I think the patterns that emerge there are going to be explained by the kind of framework I was talking about above. The first kind of concept I want to talk about is this notion of universality. So I mentioned before that when we're thinking about looking for life in the universe, we're assuming, whether we admit it or not, life has universal features. And oftentimes when we're talking about that, we are talking about chemical features, right? So this goes back to sort of the primordial soup and thinking about life as chemistry. So the question we want to ask is, what features of the chemistry of life should we expect to be universal? And if we're thinking life is chemistry, most of the features that people have talked about so far are what we call universality in biochemistry, which is the fact that the basic building blocks of life, the fact that life relies on DNA, RNA, amino acids, and certain classes of molecules that all life uses, they allow us to trace the ancestry of life on Earth to last universal common ancestor. And I'm really glad that Kate was talking about that earlier because there's a nice connection there between our talks. So it allows us to do that, but then we have this sort of idea that all life on Earth is related by this n=1 problem that Kate was talking about, that we have this, we only have one sample of life. And so we use that usually to inform, like we want to look for amino acids as a biosignature on other planets, or we want to look for oxygen production on exoplanets because life on Earth invented photosynthesis, and therefore we're kind of extrapolating the chemical details of life on Earth to other planets. But at the same time, we have this idea that universality means they should be similar in their generality, if not the details. So is there a way to generalize beyond the chemistry of life we know on Earth to what other kinds of chemistry might be possible? And for that, I want to bring in this idea of universality as it exists in physics, because it's a very different concept of universality. Universality in biochemistry, the way we talk about it in astrobiology, is universality of component parts. All life on Earth shares amino acids. So that's a universal component of biochemistry on Earth. And then therefore, sometimes as astrobiologists, we extrapolate that to being potentially a universal component of life elsewhere. And we don't always make that sort of assumption explicit, but it's kind of part of the intellectual exercise of what we're doing. In physics, universality is more of what's called a macro scale feature. So it's not a property of individual components of a system, but it's a property of how the system behaves as a whole. And this usually shows up in the physics of phase transition. So you can have two very different physical systems, like the phase transition from liquid to gas, or the phase transition in a ferromagnet, which behave very similarly. And we talk about the universality class in terms of scaling exponents at the critical phase transition. So this beta for both of these is the same. So it has the same behavior with respect to temperature. And therefore, we talk about the physics of these two very different physical systems. They have very different material properties, but their physics is universal. They're in the same universality class when we describe them in this way. And so what I'm interested in is sort of what's the universality class for biochemistry, if we can talk about that. So is our planet, with all of the biochemical diversity on our planet, a member of a universality class that we might expect other planets to also be part of in terms of the organization of chemistry on the surface of that planet that we would call life. That universality class describes living planets. And so the way we do this is actually extrapolating planetary biochemistry to networks. So you can represent biochemistry in terms of a network by having molecules be nodes and connect them with a line if they participate in a common reaction. And then you basically build up this network structure that describes the chemical reactions happening in biochemistry. And then we can use scaling laws, which are just basically some systematic regularity of a property of biochemistry changing with the size of the system to try to look for universal laws. And to do that, you need ensembles. You can't just study one system. You need to actually look for regularities that apply across many systems. And in thermodynamics, we have ensembles all the time when we talk about statistical mechanics, because when you talk about temperature, temperature is not a property of one system. It's a property of many systems with different possible microstates that all happen to share the same temperature. So we're kind of looking for the equivalent of temperature, but for biochemistry at a planetary scale. So these ensembles are built up using data from the Joint Genome Institute. The genomic data is annotated with the functions of the genes in terms of enzymatic function, and then we use that to construct what kind of biochemistry different systems are actually catalyzing. And with that, there's a lot of different ways we've been looking at the data. So we've done a lot of network analysis. We've done a lot of stuff in like cheminformatics, which is looking at individual properties of molecules like chirality and things like that. What I want to talk about today is really this idea of coarse graining chemical reaction space. So coarse graining is looking not at the details, but looking at some aggregate features in terms of the functions of the reaction. So part of our reason for doing this is that enzymes, so biomolecules, proteins that catalyze specific reactions in biochemistry, are actually organized hierarchically in terms of their function. And you can think of class EC1, all oxidoreductases, as a grouping of all possible reactions with oxidoreductase function. And so this allows a natural way of basically just looking at high-level function of biochemistry and trying to say, are there any systematic patterns? So you can think of these as kind of like temperature and pressure variables potentially for looking at biochemistry. And so the question we had was, if we look at these in this kind of macro scale view, do we see any systematic regularities that allow us to talk about potential universal properties for biochemistry that might form this kind of idea of a universality class? And to do that, we basically had to go into the three domains we had data from, and we also looked at ecosystem level data with metagenomes and counted each kind of function and then grouped them and then looked at scaling behavior. So on the y-axis in the plots I'm going to show is basically the number of functions in a particular group. So it might be the number of functions in an individual genome or metagenome that correspond to oxidoreductase, and the y-axis is the total number of functions encoded in that genome or metagenome. And what we do see is that biology does have very systematic and regular scaling behavior. So there is a statistical property here that seems pretty universal as far as how biochemistry is organized functionally. And just to look at sort of the details of the universality classes or the candidate universality classes that emerge from this, oxidoreductases are superlinear, hydrolases are superlinear, and then the transferases and isomerases and ligases are mostly sublinear. So superlinear means as the system size increases, you have an overrepresentation of that enzyme function in systems. And if it's sublinear, it means that you have an underrepresentation as system size increases. And just as a note, liases have this kind of odd behavior where they're not universally consistent with any kind of behavior, but they have very similar function to hydrolase. They both break apart molecules. So one of my students actually binned those together, and it explained a lot of our data better because they're as a class together, they exhibit superlinear behavior. And there's also some other features that are more consistent across the analysis we did if they're grouped together. So in reality, what our analysis is showing is there's really 5 universality classes. And isomerase also has a sort of statistical outlier, but it's also consistent with being sublinear. It's just better fit to linear. So it seems like there are these 5 universality classes associated with these 5 higher level functions if we combine hydrolases and liases. And so one of our questions was, is this feature of universality that emerges in a physical sense related to universality in a component sense? So can we say anything about this actually being a really good candidate for a universality class in a physical sense, because it's not directly derived from the fact that all life on Earth that we know about shares component functions? So to do that, we actually went in and looked at how for each enzyme function, how many how many of a given data set include that function. So it's basically a score of universality. So if you look at the x-axis in these plots, if it's 1, it means that enzyme function occurs in every single member of that group. So this here I'm looking at orange, which is ligases. So this enzyme function in archaea would be present in 100% of archaea. And we just rank ordered it. So these curves are actually showing the rank ordering of universality of functional components in the different classes of data we looked at. Pan-taxa is just binning the 3 domains together. And then what we see is there's sort of clear patterns, and some enzyme classes are more universal than others. So the sort of a main sort of standout is that ligases are much more universally distributed across all of our databases. So they're the orange curve here than oxyreductases. And then to sort of systematize this and quantify it so we can actually do some comparisons, we assigned an area under the curve score. So you basically calculate how much area under the curve is. If it's 1, it means it's all the way universally distributed. So it would be, you know, every enzyme function is found in 100% of that data set. So values closer to 1 mean things are more universal. And so you certainly see this with the oxyreductases. They're very not universally distributed, so they tend to be more unique functions in more in different systems than other enzyme classes. And ligases, as I mentioned already, tend to have more functions that are specific, that are common across biology. So this allows us to kind of talk about which components are more universal or less universal, not just say this appears everywhere in biology. And then once we do that, we can actually say that the universality classes are not explained by the enzyme universality, because we don't see any correlation between scaling coefficients that we observe, the actual macro scale systematic trends across the data, and the universality of the component part. So the AUC score, again, is capturing the universality component. So ligases tend to be more universally distributed. But we don't see that there's a strong correlation across any of these plots with scaling coefficient being systematically related to AUC score. Universality. So the universality in these statistical trends is actually decoupled from universality in component parts, which gives us some optimism that these might be features that are universal, possibly represent universal constraints on biochemistry that are independent of the components that biochemistry on Earth has used. And so one of the things that we want to do next, so this is some data that was added to the revision of our paper, which is going to be in PNAS soon. But it's kind of forward-looking in some sense, because one of the things my group is trying to do now is understand how to develop theory that explains these regularities so we can apply it to other chemistry. So I have this sort of vision of one day coupling sort of our network analysis, statistical analysis, and some of the stuff with assembly theory I'll talk about in a minute, with machine learning algorithms, basically evolve biochemistries that might occur in other geochemical contexts and be able to statistically predict biochemistries on other planets. But to be able to do that, we need to know the functional principles. And so one of the ways of doing that is actually looking at how the different functions are related to the actual structure of biochemical networks. And this is just showing 2 sets of data. One is for enzyme networks. So this is just doing this sort of network representation at the level of which enzymes have common substrates and reactants, and looking at the patterns that we see as far as the network structure of those features. And one of the things that we did see is that there's some correlation between some of the network properties, and in particular, sort of how local versus global connectivity is driven in the network, and whether the enzyme function is one that scales exponentially, like super exponentially, is super linear or sublinear. And it's a little bit easiest to see in an actual different algorithm that we looked at, which is something called network expansion. So if you start with just a few compounds that were present on the prebiotic Earth, and you take the sort of biosphere level network of all the biochemistry we know and is cataloged in databases today, we can actually start from those reactions and expand out into the network and track how functions were added as a function of that expansion. And you see the sublinear classes tend to contribute early to the expansion process, but they contribute a much weaker level of new functional diversity to the biosphere in sort of the periphery of the network as the network expands. But those superlinear classes are actually still contributing a lot of functional diversity, even at late times in this network expansion. So superlinear behavior probably comes from this idea of driving the actual growth of the network. And so oxyreductases obviously are doing that because life is expanding into different environments and the environments are changing over time. So that's a clear, clean example. And so to connect this back to sort of early life, one of the things that we're interested in is whether we can use these scaling laws to infer properties of the last universal common ancestor in a way that might be independent in some sense of the specific component parts of modern life. So we did compare sort of the scaling trends to what sort of a consensus model of Luca would predict for enzyme functions. That model came from Aaron Goldman. Luca's sort of within the range of scaling laws, but there's some notable differences. And these are sort of the statistical outliers versus things that actually, fit the Luca data. And so this is kind of more of a look ahead, like what we'd like to do is be able to use, if we can determine law-like properties of these scaling laws, be able to use them to build better tools for inferring properties of early life and also life on other planets that aren't dependent on the specific molecules that life is using or the specific reactions. So in the last few minutes, I'm going to talk about assembly. I know I'm running short on time and I want to make sure I have time for questions. So I'm going to go through this kind of fast, but I kind of introduced the idea already. So one of the key questions is what principles will explain life? And I feel this every day. So here's another Einstein quote. One can best feel in dealing with living systems how primitive physics still is. And the reason I put this quote here is because one of the things we're trying to do with assembly theory is actually develop a new theory of physics that applies to the properties of physics we associate to life. And as I mentioned before, that theory has to somehow exist in chemical space, at least for the emergence of life. I also think that theory exists in the space of all possible computers and cups and people and things, but chemical space is much easier to iterate because you actually can specify the combinatorial diversity and talk about it quantitatively, whereas the space of all possible tables is kind of hard to imagine. So one of the things that we're interested in with assembly theory is actually formalizing what we think information or evolution is in terms of that space of possible objects. And so in assembly theory, as I mentioned, we have an object, say like an ATP molecule. We can look at the bonds that object is composed of, and then we can start trying to assemble the object based on those bonds and look at the different pathways. And then we assign assembly index as the shortest pathway in that space, which is supposed to be sort of the minimal number of physical steps to construct that object. And one of the key features of assembly theory that sets it apart from other theories that deal with sort of informational properties, like say algorithmic information theory or Kolmogorov complexity or these other things, is that this is actually a physical observable of a molecule. And to develop that, that was actually done in Lee Cronin's lab. So a lot of the this is very experimentally motivated theory. What they did is they basically took a bunch of living samples and they looked at the molecular assembly, so that shortest path in the space, and they were able to demonstrate that that actually can be measured in mass spec. It's correlated with the number of fragmentations that you get in a mass spec. And then once you sort of go through all these different data sets, you see a threshold where there's a step size of 15 or so, above which you only see living samples. And so that 15 was actually theoretically predicted by some simulations that Cole Mathis did, where he was looking at if if you start from the basic building blocks and you try to assemble things at random, at a step size of about 15, you have less than 1 in a mole chance of making even one copy of that object. And when you go to a mass spec instrumentation, for people that work with that, you know that you need at least 10,000 copies, or you need multiple copies at least, of a molecule in order to be able to even detect any of its features. And so there is this sort of other feature of assembly, that it's not just that this object has a large depth, but you also have to have a high enough copy number that you say that there was actually a physical system present that could make a large abundance of that molecule. And this is just showing the data again, that once you get to this sort of high molecular assembly, high complexity, high number of steps, only living samples are there. So, so part of the sort of conjecture of this theory is, when you're thinking about the space of what is possible for the universe to assemble, there's a low assembly universe, which is consistent with the physics that we know and kind of describes the dead universe. When you get above this assembly index, all of those objects require information in order to exist in any abundance that you would ever observe. And therefore, it's sort of a signature of the fact that this kind of informational principles that we're talking about has taken over the dynamics of those systems, and is actually the explanatory framework for why those objects are existing. So things like proteins or DNA or cells or cups or people would all be on this sort of side of high assembly number objects. And so part of the construction of the theory now is actually to build theory that explains why that happens. And that's very deeply related to a lot of the intuition I have about physics of information, and the relation between information causation and what life is sort of a more fundamental process. So this is kind of just a segue into thinking bigger ideas. Not a lot of time to go into them here, but I'm many of our papers on this topic. And then in the last minute or so, I want to just talk about aliens and how we can look for them, because of course, as astrobiologists, we're interested in aliens. And all of this sort of deep speculation about the nature of ourselves is because I think, we're not going to, I don't think that aliens are going to kind of slap us in the face and they're going to be obvious when we see them. I think we'll get incredibly lucky if that's the case. And I think about it much more. And I think there's this implicit bias that because we are life, that will automatically recognize life when we see it. But we're also gravitational objects, and we didn't really recognize gravity, even though it was all around us for hundreds of years. And who knows if we really have the final understanding of it, but we at least have one that allows us to understand, properties in the distant universe. I think with life, we're not nearly there yet, and I would like us to get there to the stage where we're actually testing deep hypotheses about the nature of ourselves when we're looking for life on other planets, instead of just looking for the molecules of life and hoping that that will tell us enough that there's a living system there, because it would be so much follow-up work to really confirm it. And ultimately, like, really what you're looking for is an explanation of what life is. That's the central question of astrobiology. To look for it elsewhere, you have to have some ideas you're testing about what that is. So one of the ways of getting at that, I think, is actually building better experiments here on Earth that are these kind of massive statistical explorations of chemical space using some combination of the statistical laws I was talking about and sort of these assembly theory ideas. And we're trying to get at that in terms of being able to couple some of the experiments to planetary context with a NASA-funded project right now that's combining some of the stuff that geochemists do with these kind of computing chemical space type things that Lee's lab developed. And we're actually going to build this geocomputer here at ASU in the lab of Hilary Hartnett and Everest Chalk that's going to be exploring planetary context and trying to do these sort of multiple arrays of different minerals and different conditions to try to actually explore what chemistry comes out. Also, we can think about, if we're looking for life elsewhere, can we look for it in terms of molecular complexity or statistical properties of the chemistry instead of looking for specific molecules? Does that help us? Does it increase our confidence level because we actually can assign statistical likelihoods to these processes being happen? So one of the big things is, if you detect amino acid or phosphine on Venus or something, what's your confidence interval that you've actually detected life? We can't even talk about that right now. In physics, because we have these guiding theories, when you make a detection of something like the Higgs boson, you're probing fundamental physics or gravitational waves. You have a confidence interval and you have a standard for what your confidence interval is for that discovery of actually confirming that theory. And I think that would be incredibly powerful for astrobiology as far as the global dialogues we have in terms of how we communicate the detection of alien life and how we should be thinking about it. And in terms of searching for life on exoplanets, we're trying to develop assembly theory for that. And I also think that these statistical models of what the chemistry of a planet is doing are going to be incredibly useful, I hope, in the long term for thinking about how we infer the presence of life on exoplanets in the presence of limited data. And with that, I'm going to end with one of my other favorite quotes, which is really telling about the physics I'm interested in understanding, which is that base metals can be transmuted into gold by stars and by intelligent beings who understand the processes that power stars and by nothing else in the universe. And so when we say, are we alone and we're looking for ourselves, I think we are looking for a very deep kind of physics about the nature of our reality and what we are in it. And I really want us to get there so we can understand more about ourselves. And so with that, I'm going to thank my amazing research group. I have absolutely the best research group in the world. They're so smart and intelligent. They push me super hard. And also all our funders and collaborators and all of you for listening. And I'm going to wrap up there and hopefully still have enough time for questions. Thank you.
Incredible, Sara. Thank you so much for a beautiful and thought-stimulating talk, and also for the interesting perspective on astrobiology itself. I myself didn't realize how young this field is. Now I feel really young. Well, I have some questions
for all of us.
Oh, yeah. Yes, so we have some questions. I will read them in a moment. I will allow myself to ask my question first. You mentioned that scales are important. For example, you said that gravity is not that important on a nuclear scale, right? So would you imagine a life on different scale than the one that we know, or is it not enough complexity or interactions?
Yeah, I thought about this a lot. So I think the way I come at that question is, like when we're thinking about the sort of scales of physical reality, right? You go all the way from subatomic physics all the way up to like galactic scales, and different physics is dominant at those scales. But we have this sort of intermediate regime of complexity, which is like, from chemistry all the way up to like technology, right? There's like this band of this high density of things happening. And it could be observational bias that we're seeing so much happening at that scale because we're here. But I think really the way I think about it is chemistry is the first scale of physical reality where you actually get combinatorial diversity mattering, right? Like when you're at subatomic particles, there's a certain number of them, but they don't do much interesting by combining and forming higher level structures. But when you get to chemistry, that starts to become a really important process, and you do get this combinatorial explosion. And so I think this physics emerges there for a reason. So I think the origin of life is in chemistry because the physics of life emerges in chemistry. That's the first scale of reality it's relevant. But I do think that also that it's just as much manifest in things like technology or silicon chips or rockets as it is in chemistry. Does that answer your question?
It does. Thank you. Yeah. One more comment is that you were talking a lot about possible protein functions and how common they could be. This goes pretty nice in line with the research that we're doing right now, that we try to assemble simple abiotic peptides to build higher order structures, and we look what kind of functions they could do. So I think I could use some of your work to specify what kind of function we're looking at. We're looking for, right? So cool. There may be more likely to emerge. So very useful for me too. Thank you. And now going to chat to questions from chat. First question is: do you think that inorganic life could form on some planets? If yes, do you think such life needs information polymers to enable self-replication?
Yeah, it's a really great question. I think, so again, like sort of the picture that I'm interested in is life is not a binary thing. It's not this is life and this is not life. It's actually more like a continuum of processes. So some systems might have more life in them than other systems, more information in them. In the same sense, like a black hole has more gravitational potential than the Earth does. Like it's a deeper potential well. So from that perspective, I think inorganic life can exist. It would just the more interesting question there is, does it have the potential for open-ended evolution to become more life, right? And so there is this sort of critical transition that at least biology on Earth underwent, where we sort of cascaded into what appears to be open-ended novelty generation, and that's sort of one of the most mysterious features of life on Earth if you look at it over the course of billions of years. And that was in part mediated, as the second part of your question, by informational polymers. And I do think whether it's a linear informational polymer or not is sort of an open question, but I do think that you need something that retains memory of past states and copies them into the future. That is an essential component of that transition to open-ended growth and complexity. Because if you can't remember how to build things, you're not going to be able to build them again. So there's probably some primitive inorganic life that might exist, primitive, just simple examples of inorganic life in the universe that have this sort of, causal structure allowing them to build complexity. But unless you have better information storing mechanisms, you're not going to get to those sort of subsequent transitions. And this is consistent, say, with like the major transitions in evolution hypotheses that people have put out, like Zathmari and Maynard Smith, about information storage mediating many major transitions in evolution. It's just kind of looking at it from a much more abstract perspective.
I understand. I think very well in line with this question comes another one that says, is there a possibility for life forms to be based on alternative biochemistries?
Yes. I don't see any reason why not. And I think part of the issue is, chemical space is, like I said, exponentially huge. And the amount of it that's been mapped by biology and technology on Earth, like all the reactions and molecules we know, is like a very infinitesimal fraction of that space. So there's a huge industry of people in cheminformatics, particularly in the drug industry, just trying to predict properties of molecules and actually understand, what kinds of molecules could exist. So we don't actually really know much about chemical space. And so people make a lot of sort of arguments that, well, there's special properties of the biomolecules we know, and we don't see those properties generically in other chemistry we know about. So therefore life must use these molecules. But I think, there could be other islands in chemical space out there somewhere that we just haven't explored that have all of those properties. That's a little bit more speculative, but also so yes, I think life can use other chemistries. I think the main thing I'm interested in is sort of like, what are the system level properties, so we can predict which other chemistries are possible that maintain those system level properties.
Okay, we're getting a lot of questions and a lot of thanks to you for a very stimulating, visionary even talk for Sam. And so I don't want to miss any of the questions, so I'm not going to gather them by the subject anymore, but just read from top to down.
Sure.
So the next question is, what signatures for life do you think we should look for in data from upcoming observation missions, example from James Webb Telescope?
Yeah, so JWST is a little harder. I think we're a little ways out from really doing rigorous biosignature searches on exoplanets. I know people have a lot of candidates like oxygen and methane and things, but I really think that we have to look for more of these system level patterns, because we're not going to. I would immediately say assembly of molecules is the best biosignature we have right now. Like, that's my bias. I've worked on that theory. I understand like sort of its connection to the deep physics pretty well, and it's observationally testable, which is why I'm so excited about it. So in the solar system, I think that's a really good biosignature and should be flying on missions. And we are talking with people about developing it for missions. But in exoplanets, most of the molecules that you have contact with in the atmosphere are very low molecular weight, very small molecules, like CH4 and O2, and they don't have very high complexity. So there's this observational constraint you get for exoplanets about, what's a volatile molecule that can appear in the atmosphere that you can detect from, tens of light years away. So under that constraint, I think we need better inference tools. And my group's been working on how to infer the system level properties of an atmosphere, like using network analysis and things like that. And we're starting to try to look at assembly and whether there are sort of observational signatures of the structure of networks and the assembly structure of molecules that actually appear in the spectra that you can infer directly from the spectra backwards. But that's like work in progress. So for me, those would be better approaches to biosignature science, because they're more complexity based, more thinking about the fundamental physics that we're actually trying to discover. But again, my bias. So yeah.
Totally. Okay, another question is, what are your thoughts on Drake's equation? How useful do you think it is? And do you think we will ever be able to reliably fill in the variables?
Right. So I think that Drake's equation was an excellent insight into organizing our ignorance. And what I mean by that is it was like it's a really great way of saying what are all the probabilities of the different processes and breaking them down. So you could talk about each one individually all the way up to technological civilization. So before that, people were just, they didn't have a systematic way of thinking about that problem. And when Drake wrote the equation down, we didn't know a lot of the early terms in the equation, like the rate of, planet formation and Earth-like planets. And those are now pretty tightly constrained. So you can see where we've made a lot of progress. But I don't think that we can actually usefully use it to predict the likelihood of technological civilizations because we haven't constrained those other terms. And so like the origin of life is unconstrained. The evolution of advanced life is unconstrained. The lifetime of technological civilization is unconstrained. And there's a lot of sort of speculation about what those parameters might be based on, sort of ad hoc assumptions about how we can generalize beyond life on Earth. But I do think the idea that there is a probability we can assign to those processes is really significant. And so what I would say is that when we have like the right theory and the right experimental tests and observations to constrain those probabilities, then we can use it to really extrapolate at the level that Drake envisioned when he was writing it down. But just right now, it's just too unconstrained. But I think it was brilliant for being the first like real probabilistic approach to thinking systematically about the likelihood of life.
Thank you. Another question is, if I understand, I'm just going to read it. If I understand correctly, one implication of your work is that sufficiently long chain hydrocarbons must be biogenic, with empirical backing from an analysis of the molecular products on Earth.
So there is an issue, so it depends on how many repeating units they have. So in assembly theory, like you don't count repeating units, like they don't get double counted. So large molecules with a lot of symmetries that are basically like really easy to decompose into parts systematically, and they end up having very short pathways. So you have to be really careful. Just because it's a macromolecule doesn't mean it has high assembly. And this is sort of one of the subtleties of assembly, like the way it actually captures causal structure and like the simplicity or difficulty of building an object. And so it is capable of distinguishing complex chemical mixtures from something that had to have a stepwise evolutionary process acquired information to build an object. And that's something that we haven't been able to do before, because of course, like if we look at Murchison or something like that, it's like, a complex chemical soupy mess. But Murchison has very low assembly when you actually put it through this method.
Okay, thank you. And now is the question, how do you exclude some possibilities? First, abiotic mechanisms may generate these molecules, but at rates which are dwarfed by the biogenic flux and hence not detected. Do you want to answer the first part now first?
Yeah, sure. So there's a really important but subtle point about this complexity threshold that I think people it takes a little bit of like conceptual work to really get at. The easiest way I can kind of get at it simply is to say you wouldn't expect a cell phone to emerge from the geochemistry on Mars, right? This object is too complex. So we all kind of intuitively admit that there are some things that are too complex for planets to build in a non-steered, non-evolutionary process. The question is, does that threshold exist in chemistry, or is it at some higher level of organization? And what we're arguing in assembly theory is it does exist in chemistry. Some molecules are biosignatures or even technosignatures. They could only be assembled by technology. If you think about drugs like Viagra or Taxol, like those molecules are so complex, biology can't produce them. So the argument is there are some molecules geochemistry can't produce. It's not going to produce, say, a macromolecule protein of 500 amino acids with specificity and high abundance. And so it might be that you could get a one-off random fluke somewhere in the universe where you make that 200 length protein, but that process isn't reliable. It's not repeatable. So if there's that geochemical environment, it would have been a tail end of a distribution. It would have been one molecule that existed and just fell apart. Kind of, so that's sort of like the level that we're looking at it. So the argument is there should be a threshold in chemical space because it's so huge. And if you have a non-directed process, you're going to be basically producing all the possible combinatorial variations. You're not going to get one in high abundance. And so that's sort of the key argument. So it should be robust against false positives based on that conceptual foundation. And of course, you have to go and empirically validate that. So the assembly theory mass spec paper was the first empirical demonstration of that, but there's more work to do. So the second question about geochemical environments having different variation. Yes, we expect the structure of the assembly space to look different and the kinds of molecules produce different in different geochemical environments, but we still expect that general argument to hold because it's at sort of a much deeper level. And I think, you could always make the argument, well, what if I had the pH just right and I had the the minerals were just so, and the temperature was fluctuated here like this. But now it's really difficult to see this because we do this in prebiotic chemistry when we design experiments. But now you're putting your intelligent agency into building all those steps. And then you have to ask, what's the likelihood of the universe producing each of those steps and doing it reliably? And how much information is necessary to specify each of those steps? And what you find is when you get to high assembly molecules, it's impossible unless you have a physical system that has those constraints in it, which means it has the design of the object or the information of the object. So if you want to argue against assembly theory, it's kind of saying that chemistry has design of biomolecules in mind, which I don't think most of us want to argue. It's kind of formalizing that intuition in a really rigorous way and trying to make it experimentally validatable. I can't hear you now, Tomas.
Sorry, thank you very much for explaining it. It is actually something I have to put some thought into to start understanding. Another thing is, in the question number 7 that I have here is if the live or non-live distinction is more of a continuum, do you still subscribe to the interpretation of the origin of life as a phase transition phenomenon? So the question about phase transition, and does molecular assembly has a say this topic of phase transition phenomena?
Yeah, this is a great question. I go back and forth about whether I think of the origin of life as like a gradual transition or an abrupt transition. But I think, some of the ways I conceptualize about it are a little like the quantum to classical transition. We know it's there, but like mapping the exact boundary is kind of hard because we're talking about different physics. And I think about the origin of life that way. There's some transition that mediates that some physical systems can move past this assembly boundary. And that's certainly some kind of, it is a phase transition, but it's not like a canonical physics phase transition because it's actually a transition in the causal structure of like the physical systems themselves and how they actually are related to other physical objects. So I think molecular assembly has a lot to say on that. And that's actually one of the things that I'm working on quite a lot is to bring. So I talked a lot earlier in my career about ideas about top-down causation and information being causal and all these kind of things. What I'm working on now, like as being key features of life. So top-down causation is this idea, I can have a thought and move a cup. It's like some kind of top-down, like from my mental states to the physical world. And top-down causation seems to be this kind of pervasive feature of life. But it's very hard to explain that feature in terms of current physics to the point that people just assume that feature doesn't actually exist and all causation stems from microscopic level physics. In assembly theory, that's not actually true because every object is a unique cause that exists in the universe, and then and all of its causal structure is embedded in its assembly space. And so you can feasibly talk about the information and causation in an object in a really rigorous way. And then when we do that, I think there are some phase transitions that happen in the structure of that space that we would associate with the transition to life.
Thank you.
I know it's very abstract. Who would have thought, right? I'm like, life is abstract. It's very abstract.
Yes, absolutely. And the last questions we have here, it refers to the simplicity of life.
Nice.
So there is a little bit of introduction that I'm just going to skip, and you can read it here on the screen. But is there anything special in chemistry not present in a virtual environment which pushes biology evolution towards super complex life forms?
Yeah, so this is an excellent question. I think it's kind of getting at the difference between artificial life and biological life, right? So people try to simulate evolution in the computer all the time, but they have a really hard time with this open-ended evolution problem. Like, how is it that continual novelty is generated? Most evolutionary algorithms flatten out at a certain point. I think this is kind of what they're getting at. So I do think there's something very different about the physical implementation, and that has to do with the fact that at least the way we think about it in assembly theory is all physical objects are in assembly space. So you see me right now as like an object Sara, or you see a molecule as like CH4, but the way it actually interacts in assembly is as this space of the possible ways of assembling that object. Like all of that causal structure is embedded in the object. And for molecules, that's actually an observable property of a molecule. In the same sense, the charge of an electron is an observable property of the electron. And so it's actually the structure, it's the causal structure of the spaces that's generating the novelty. And the one question is the universe actually generating genuine surprises, or is all of it predictable based on the structure of that space? And I think that's a question that we're really grappling right now. So like those of us working on assembly theory are talking about this a lot. And I think part of the, we don't have a mechanism for understanding that the universe could possibly generate anything entirely new that's not predictable based on past things that existed. And it might be the case that actually is what the universe does. And so that's also kind of a really weird feature. But I'm still optimistic that most of the information is actually embedded in the assembly space. And it's just a matter of understanding the properties of those spaces, because they're giant causal graphs, right? And now you're asking, like, what are the features of those graphs and how they interact in order to generate new things? And so it's a computationally complex problem as far as how we can structure that. And we're doing a lot of statistical models and things to try to get at that, but it's challenging. So, so my short answer, that was a long answer, but the short answer is yes, things that physically exist have properties that we are not encoding in our computational models right now, or simulations, that are actually driving open-ended novelty.
Okay, so maybe a little bit provoking question as the last one is, what is your gut feeling? Is life common in the universe?
Oh, yeah. I am optimistic, but I think as an astrobiologist, right? Like I want to understand what we are. So I want to understand if there's other things like us. But I don't know. I don't like scientifically have any kind of window into that. And there are ways of structuring thinking about that, but there's still a lot of assumptions, and even in assembly theory. Like I could say, if I count the assembly of the universe and say we have a principle in assembly theory of assembliness increasing over time, there's spatial degrees of freedom, and then there's what's happening on our biosphere and our planet, and like how much of that is actually on one planet versus in all of the universes. And there's people that work in cosmobiology stuff. There's like a whole group of people trying to think of degrees of freedom separate from assembly theory that do that, which is pretty cool stuff.
Absolutely. And I think one of the comments that showed up on chat very nicely wraps it up, that your talk reminded us to be more careful when we're using common terms such as life in scientific discussion. So thank you very much.
All the undefined terms.
So thank you so much. It was a beautiful presentation, a beautiful discussion at the end. And I will be closing this session for now, and we'll see each other tomorrow. Actually, today we still have a poster session, so please visit our posters. And tomorrow we will start with 2 parallel sessions on prebiotic chemistry and planets and space exploration. And then the second couple will be origin of life and evolution, and human space exploration, including space medicine. So thank you again, Sara. Incredible pleasure.
Thank you all. It was great.